me go ahead. Okay, so I'm actually gonna talk about gene regulation in relation to cancer development, how a cell undergoes malignant transformation from a normal cell to a cancerous cell. And when so this is definitely, I'm assuming, over an hour and 15 minutes. So I'll either have an extra credit question about cancer on the exam, or an assignment, extra credit assignment, or if you want to watch because you're just so interested. Okay. So you're going to see my cynicism emerge about how society and the scientific community views cancer. I don't want to go on too much of a rant or ramble on here, but the textbook version is sort of, it's the official definition and understanding of cancer and what defines a cancerous cell, the origin of a cancerous cell. How does a normal cell become cancerous? That's in the text, and we're going to talk about it. But I was exposed to a just remarkable scientist at my undergraduate university. And I think he's doing groundbreaking work with really revising that understanding, improving upon it. And so I'm sort of, it's just very interesting. And, even in this lecture, I've mentioned a few times that, oh my gosh, we never used to think, that, like Lamarck, for instance, we never used to think that Lamarck actually had some truth to his idea that if I went to the gym, decided to go to the gym every day, just eat a bunch of protein and just get big, that might affect my offspring. No, we thought it had to affect the DNA sequence, germline, that DNA, we had to affect the actual nucleotide sequence. But we now understand that that's not entirely the case. And so, as a scientist, biologist, I love to question, to doubt, to explore in order to reach the most, deep and comprehensive understanding of what we're talking about. So without going too much upon this path, let's talk about what the textbook talks about. The official textbook version of cancer gene regulation, winky face, or lack thereof gene regulation, right? So first, let us sort of identify the stressors or the insults that are going, that are going to, cause this cancerous transformation in a cell. So stressors that, first I should mention really quickly, or maybe what causes cancer? Well, carcinogens, radiation, viruses, inflammation, certain genes like BRCA1. What are all these things doing to our cell? That's sort of how the textbook and how scientists originally came up with this hypothesis of what is causing cancer in people and cells. What's making a cell cancerous? Well, it's these stressors. What are they doing? What does radiation do? What do certain viruses do? What do like Rouse sarcoma virus and papillomavirus, ovarian cancer, all these certain viruses, hepatitis virus can contribute to liver cancer. So anyway, basically, okay, just had to double check something. All right, so stressors that, what do? They cause DNA damage, induce mutations. You see mutations, changes in the DNA sequence from these types of external stressors. So stressors that induce DNA damage. Carcinogens, so cancer-causing chemicals. Radiation, like ultraviolet from the sun. Inflammation. Viruses, certain inherited genes like BRCA1. Call it an oncogene, but we'll define that shortly. So in the lab, we've exposed cells to these things. We expose mice to these things. And then we look at their genetic sequence and, hey, we just caused a bunch of mutations. The DNA changed. Its sequence, its order of nucleotides changed. And we know these things are associated with cancer, right? People in the sun too long, skin cancer. These viruses cause cancer and they're affecting DNA, integrating in a host genome. Exposing chemicals to cells will cause these cells to exhibit these cancer-like characteristics. So that's what we think cause it. So what's happening here is that what we call proto-oncogenes. Proto-oncogenes these are normal genes that can become oncogenes. So the proto-oncogene is the normal version, but they can become oncogenes. These are the cancer-causing version. So cancer-causing version via mutation. So what are these oncogenes typically? Well, cancer has a set of hallmarks. Cancer cells divide rapidly, uncontrollably. They migrate, they metastasize, they move everywhere. They grow on top of one another. They're pretty much undifferentiated. They all look very similar. They're amorphic, sort of. But really, the key hallmark here is that they're growing and dividing like crazy. Unregulated, uncontrolled cell growth is kind of the definition of a cancerous cell. And so these oncogenes typically code for proteins that stimulate cell growth and proliferation, which is rapid cell division. Okay. Okay. So an example textbook talks about is RAS. It's an oncogene, RAS. It's involved in a signaling, it's a receptor protein that when it's not working properly, it can be activated uncontrollably. Just constitutive activation without even a signal. And essentially the downstream effect of this RAS receptor is excessive, well, cell growth and division. Its downstream gene products are involved in cell growth and division. Okay, so the next category of genes involved in cancer development, they're called tumor suppressor genes. And tumor suppressive genes, they actually serve a variety of roles, protective roles in the cell to prevent uncontrolled cell growth. So let's, don't really have to write that. Let's just write those roles. It's 3 well-known ones. So certain tumor suppressor genes repair damaged DNA. Or rather, the proteins that these genes encode for are DNA repair proteins. So they're going to repair damaged DNA. Why would that be good in preventing cancer? Well, if cancer arises from damaged mutation or mutations in DNA, damaged DNA, then repairing that DNA before the damaged DNA accumulates and is passed on to the next generations of cells would prevent cancer from developing. The another role is adhesion, cell adhesion. So certain tumor suppressor proteins are involved in adhesion to other cells or the ECM, which stands for extracellular matrix. So normal cells, when they're attached to other cells or this extracellular matrix, which is this network of fibrous proteins like collagen and a lot of carbohydrate groups as well, and proteins that are glycosylated. And by the way, the ECM, you don't really have to know what it is, but it surrounds cells, it surrounds all of our organs and tissues. It's sort of the in-between jelly substance of sorts, but it does provide structural support and allows for elasticity. So certain organs like the lungs, the skin, the ECM has more of these elastin proteins that allow for flexibility. So importantly, proper cell adhesion signals to cells to stop dividing and growing. So cancers exhibit what we call anchorage independent growth. So they can be attached to other cells, but they're still growing. So that signaling is not working. That something's going wrong with that. Okay. The last role for tumor suppressor genes to prevent cancer is that they can be components of cell signaling pathways and these pathways actually inhibit the cell cycle. So prevent the cell cycle progression and then going to division, right? So let's just write tumor, or actually, sorry, let's just write proteins that are components of cell signaling pathways that inhibit the cell cycle. So let's take a look at an example. Of a tumor suppressor gene. This one I just, had to teach you about P53. It's just very, very well studied and famous tumor suppressor protein. In fact, it's kind of interesting. They found, so we have one P53 gene, and they found that in elephants don't get cancer very often, but they have so many more cells, so much more cell division and possible mutations that can accumulate. So they wondered why. And of course, the textbook admits there needs to be more investigation into this, and I agree. But they do have like 20 copies of P53. So that's pretty interesting. Anyway, so a signaling pathway that starts with DNA damage leads to the production of this P53 protein. And it's a transcription factor. P53 is a transcription factor, and its target genes are going to be directly involved in so activating, by the way, we're almost done here. I was just looking at the rest of my notes. Been a long lecture. So activating genes that are directly involved in DNA repair. That's the first function of P53 as a transcription factor. Second, if the DNA damage is too extensive and it can't be repaired, P53 also can activate genes whose protein products trigger apoptosis. Apoptosis is the term for programmed cell death. It's a very organized degradation of the cell. So importantly, this is it. So importantly, so P53, they find that over 50% of cancers have P53 mutations. But that begs me to ask, okay, what about the 49% that don't? They have functional P53. Why are they still cancers? I don't know. So, well, you know, that's me asking questions. So essentially the idea is that with damaged P53, you're not going to get these DNA repair mechanisms going, and you're going to accumulate these mutations more rapidly. The cell passes them on through successive divisions, and then cancer results. Now, let me show you. So the multi-step model of cancer development. So quote from the textbook, the development of a malignant tumor is paralleled by gradual accumulation of mutations that convert, I should say, proto-oncogenes to oncogenes and knock out tumor suppressor genes. Knockout meaning rendering them non-functional. So any biology textbook, that's the definition of cancer. That's what causes cancer, mutations. And so the advent, if you've heard of personalized medicine, that's a huge thing. And so breast cancer, I worked in a lab, and my boss, the guy who runs the lab, was part of a team that discovered HER2, it's a very famous therapeutic marker in breast cancer patients. Really a needle in a haystack, unfortunately, and a lucky find by, you know, in my eyes, because so many billions of dollars, so many scientists, so many labs, so much research into finding therapeutic markers hasn't yielded much since. And even that one, I'll never forget my boss saying, you know, with pride, sadly, because the cancer field is so abysmal with progress, unfortunately. But, you know, 25% of breast cancers have HER2 mutations. Of those, half respond to Herceptin, the treatment. So even if you have the genetic mutation, the HER2 mutation, the treatment might not even work for half of them. I was like, what? That's not very good. And he lives in a multi-million-dollar mansion in Pasadena because each Herceptin pill is $7,000. It's just pretty sad. And so, you know, I was lucky to be exposed to this other scientist research, because I at the heart of it, I'm not too cynical. I mean, I get, you know, money talks and it's a big business with cancer, but I'm not as upset about that or interested. I'm more interested in the actual science, and the actual science that this other guy is doing is quite just so cool and innovative. And I parallels a lot of Nobel Prize winning discoveries. But this is the multi-step model that I just mentioned. It's so I like to include it because it's really, I think it's not very specific. And as scientists, we like to be precise. We like to have, you know, the complete picture in a very detailed, accurate manner. And so you've got loss of tumor suppressor gene, you start to get this benign thing, and then another gene you get activated, oncogene, loss of another tumor suppressor gene, loss of another tumor suppressor gene, and then 5 is additional mutations. This is from the textbook. Like, to me, that's like, what? That's so not specific. Which mutations? And, you know, and what about my skin cells that are loaded with mutations from the sun? But I don't have skin cancer. They have more mutations than some cancers. So there's a lot of things about, you know, the development of cancer that I question now. And that's, you know, as a scientist, it's good to be curious and question. But that's it. Oh, this is the sort of breast cancer thing I was mentioning a little bit. There's different types depending on their genetic profile. So the whole advent of personalized medicine is to personalize and tailor a specific therapy to someone based on their genetic sequence. So yeah, my grad school project dealt with this, the triple negative breast cancers. They don't have any therapeutic targets. So I was trying to find another needle in a haystack of 3 billion nucleotides. Okay, anything else I wanted to say? Oh, I did want to sort of like, just to complicate things even more and just really just make it too insane is epigenetics. Your mutations genome sequence might not matter at all. As we discussed today, you can have the mutation, but it can be silenced epigenetically. It can be methylated, this gene. So the oncogene or it's not expressed. And so you don't develop the cancer. It's, you know, so it's, anyway. All right, that's it.